You can't see it, you can't smell it, and by the time you know it's there, it's already too late. Welcome back to Cell Culture Series, and today we're talking about the thing that every researcher dreads, but most don't take seriously enough until it's already destroyed their experiment. Contamination, the silent killer of cell culture. Studies estimate that between 15 and 35% of cell culture experiments worldwide fail, not because of bad science, not because of poor hypotheses, but because something invisible crept into a flask and sabotaged everything. Today, we're going to know our enemy. We're breaking down every type of contamination, how to detect it, how to prevent it, and the catastrophic scientific scandal it caused when no one was paying attention. Let's build our contamination mugshot board. There are 4 main suspects, and they are very different criminals. Each one requires a different strategy to catch, and some of them are far more dangerous than they look. Our first suspect is the most obvious offender: bacterial and fungal contamination. This one announces itself. Within 1 to 3 days, your media will turn turbid, visibly cloudy. The pH indicator in your media, phenol red, will drop, turning yellow as bacteria metabolize nutrients and produce acid. You might see floating particles, a ring of fuzz around the edge of the flask, or in serious fungal contamination, clearly visible colonies. How does it get in? Poor aseptic technique, an uncapped flask, a contaminated pipette tip, an unsprayed surface. This is the contamination that punishes carelessness, and it hits fast. Now, Mycoplasma, this is the one you should be most afraid of, and most researchers don't even test for it. Mycoplasma are the smallest known free-living bacteria, and they have a trick up their sleeve: they have no cell wall. That means they sail right through the 0.22 micrometer filters you think are protecting your media. They're invisible in your media. Your cells look perfectly healthy. Everything looks normal, but under the surface? Catastrophe. Mycoplasma attach to the outside of your cells and directly interfere with gene expression, cellular metabolism, proliferation rates, and, critically, how your cells respond to drugs. This means if you're running a drug efficacy screen, a cytotoxicity assay, or any gene expression study with mycoplasma contaminated cells, your data is fiction. Here is the most alarming statistic in all of cell biology: 15 to 35% of published cell culture research has used mycoplasma contaminated cells. That's published, peer-reviewed research. Contaminated, and the authors had no idea. How do you catch something invisible? You have to go looking for it. The 3 main detection methods are: PCR, the gold standard, high sensitivity, uses the genetic sequence of mycoplasma DNA; ELISA, an antibody-based assay, slightly less sensitive but widely available; and fluorescent staining, using Hoechst or DAPI dyes. In a healthy culture, these dyes only stain the nucleus. In a mycoplasma-infected culture, you'll see extra fluorescent material scattered outside the nucleus, the mycoplasma lighting up between and around your cells. It's unmistakable once you know what you're seeing. Third on our mugshot board, chemical contamination. This one is sneaky in a completely different way. Your cells won't die dramatically. They'll just behave strangely. The main culprits? Endotoxins, breakdown products from bacteria that can survive on poorly cleaned glassware even after autoclaving. Residual detergents from washing bottles, even tiny amounts can disrupt lipid membranes and signaling pathways. And heavy metals from water that isn't cell culture grade. Tap water. Tap water treated with a standard filter but not proper ultrapure filtration. The rule here is simple: use cell culture grade water only. Always, no exceptions. And our fourth suspect: cross-contamination. One cell line accidentally contaminating another. On the surface it sounds almost harmless. They're still cells, right? But here's the problem: if your HeLa cells are actually MCF7 cells because someone worked on both lines in the same hood without proper decontamination, every single result you generate is from the wrong cell line. This isn't hypothetical. It happened, at an extraordinary scale. We'll tell that story in full at the end of this episode. But here's the takeaway now. Authenticate your cell line with STR profiling. Short tandem repeat analysis gives your cell line a genetic fingerprint. If it doesn't match the database, you don't have what you think you have. Knowing your enemy is only half the battle. Let's get practical. I'm going to walk you through 3 things. The Mycoplasma PCR detection protocol. The non-negotiable sterile technique habits that prevent contamination from ever entering your culture, and proper decontamination when things go wrong. Now let's talk about sterile technique, the first line of defense. Rule 1. Spray everything. Before anything enters the biosafety cabinet, it gets sprayed with 70% ethanol and wiped. Flasks, tubes, pipette boxes, your gloves. Everything. Rule 2. Never leave caps off. A flask or bottle with its cap removed is an open invitation. Work quickly. Cap immediately. Never rest an uncapped flask while you're doing something else. Rule 3. Always use filtered pipette tips. The filter prevents aerosolized particles from the pipette barrel from entering your sample. This is especially critical when working with viral vectors, mycoplasma positive lines, or shared equipment. Rule 4. 1 bottle per experiment. Never take your media bottle, the one that feeds 10 experiments, and directly pipette from it into your flask. Aliquot. Use, then discard. Shared bottles are a contamination highway. When contamination does happen, and at some point, it will, your decontamination response protocol needs to be immediate and thorough. Liquid biological waste. Add bleach to a final concentration of at least 10%, let sit for 30 minutes minimum, then dispose as liquid waste per your institution's biosafety protocol. Solid waste. Flasks, plates, tips, autoclave at 121 degrees Celsius for a minimum of 30 minutes. Do not skip this step. Do not put contaminated flasks in the regular trash. Your hood. Decontaminate the interior surface with 70% ethanol, followed by UV irradiation for 30 minutes. If you suspect mycoplasma, consider fumigation with formaldehyde vapor. Follow your institutional biosafety guidelines for this. And critically, document everything. Date of detection, cell line affected, type of contamination suspected, actions taken. Your contamination log is your lab's institutional memory. Let's do a rapid-fire mistake clinic. These are the 4 habits I see most often that lead directly to contamination, and the thinking errors behind each one. 1. Antibiotics protect me from everything. Penicillin and streptomycin kill bacteria by targeting cell wall synthesis. Mycoplasma has no cell wall. Fluconazole targets fungal ergosterol. None of these touch mycoplasma. Using a penicillin, streptomycin cocktail gives you false confidence while mycoplasma corrupts your data silently. Antibiotics mask bacterial contamination too, which delays your detection of a problem that's already there. Best practice. Grow antibiotic-free, test rigorously. Use antibiotics only when absolutely necessary. 2. Sharing media bottles across experiments. 1 contaminated flask, 1 dip of the pipette into the shared media bottle. Congratulations, you've now potentially contaminated every experiment that bottle touches. Aliquot your media into single-use working volumes. A 50 milliliters conical per experiment costs pennies compared to losing weeks of work. 3. Using cells without mycoplasma testing first. You received a new cell line. It came from a reputable source. The cells look great, you skip testing and go straight to experiments. This is how mycoplasma propagates through labs. Test every new cell line before it touches your incubator. Test your existing lines every 1 to 3 months. If you collaborate and share cells, test them before use, every time. 4. Not having a contamination response protocol. You find contamination, you panic, you're not sure what to discard, what to bleach, who to notify, or how to trace the source. A written contamination response protocol eliminates panic. It assigns responsibility, defines disposal procedures, and requires incident logging. Your lab should have one. If it doesn't, write it this week. It's 1951. A cervical cancer sample is taken from a patient named Henrietta Lacks at Johns Hopkins Hospital. The cells are given to George Gay's laboratory. And they do something remarkable. They survive. They grow. They double every 24 hours. They become the world's first immortalized human cell line, HeLa. HeLa cells were extraordinary. They were distributed to labs around the world. They helped develop the polio vaccine. They contributed to cancer research, virology, genetics. For decades, they were the workhorse of human cell biology. But here's where the story turns dark. HeLa cells are, biologically speaking, aggressive. They grow faster than almost any human cell line. And in the open, shared lab environments of the 1960s and 70s, where cells were passed between researchers, where shared equipment was the norm, where biosafety procedures were primitive by modern standards, HeLa cells began to contaminate other cultures. Not dramatically, not obviously, silently. By the 1970s, cell biologist Walter Nelson Rees began systematically analyzing cell lines from biorepositories. What he found was alarming. Dozens of supposedly distinct cell lines, prostate cancer cells, breast cancer cells, lung cancer cells from different patients, were, at the genetic level, all HeLa. The cells had been cross-contaminated, and then passaged, and then published. For decades, researchers thought they were studying prostate cancer, they were studying cervical cancer cells. They thought they were modeling a specific genetic disease, they were studying a completely different genome. Today, databases like ICLAC, the International Cell Line Authentication Committee, maintain a registry of over 500 known misidentified cell lines. Research papers based on those lines number in the tens of thousands. The lesson of HeLa is not to vilify anyone. It's a lesson about systems. When you assume your cells are what they say they are, you're trusting a chain of custody that can fail at any link. Verify, authenticate, every time. Alright, let's close with the 3 things I need you to walk away knowing cold. 1. Bacterial and fungal contamination is visible, Mycoplasma is invisible, and far more dangerous. Don't let clear media fool you. Clear media with compromised cells is worse than turbid media you can detect immediately. 2. Test for Mycoplasma before any experiment, every single time. Not when you feel like it. Not every few months. Before every experiment, especially with new lines or lines received from another lab. Your data is only as trustworthy as your cells. 3. Cross-contamination has corrupted decades of published science. Authenticate your cell lines with STR profiling. Use the ATCC or ECACC database. If you can't authenticate, you can't publish with confidence. Cell culture is foundational to modern biomedical science. The treatments being tested in your lab today could become the medicines of tomorrow. That work is only worth something if the cells generating the data are what you think they are, doing what you think they're doing. Contamination is preventable. With the right habits, the right protocols, and the right mindset, you can eliminate it from your lab entirely. If you found this episode useful, subscribe and hit the bell. Next episode, we're going deep on cell passaging and the dangers of over-passaging, including how genetic drift silently changes your cells over time. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.